Hello, this presentation is going to serve as a summary on the work of separating dyes using BIST, which is a new multi-purpose separation technology developed here at SIELC. This presentation is going to follow this general structure. First, we'll be going over a brief introduction into BIST. Then we're going to go over an introduction into chemical dyes and why means of separating them are important. And then we'll follow up by giving examples of using BIST to separate dyes. They'll be separated on the basis of either their acidity, their basicity, or the count of their charge. The separation of monocharged and multicharged molecules is a recurring challenge in chromatography, and even the existing tools within mixed-mode chromatographic methods and ion exchange chromatographic methods are not universal in their ability to separate all multicharged molecules. To that end, it is noteworthy that if one were to use an ionically charged stationary phase and introduce a doubly charged ion to the mobile phase that has the opposing charge to the stationary phase, that those doubly charged ions could effectively coordinate to the stationary phase surface and switch the charge of the stationary phase's behavior. We've shown this down below in the example where we're using a stationary phase whose surface is negatively charged, and the doubly charged ion that's been introduced into the mobile phase is positive in nature. This effectively switches the stationary phase charge from negative to positive. It is noteworthy that these doubly charged ions can effectively act as bridging ligands and thus enable retention of analytes that have the otherwise same charge that was initially seen on the surface area of the stationary phase. It must also be noted that this trend can only occur if there is low water concentration in the mobile phase. If there is a great deal of water in the mobile phase, the doubly charged ions are going to be effectively solvated, and coordination between them and the surface of the stationary phase will be prevented. There are conditions of control for the retention of multi-charged molecules when using bridging ions such as these. 1. If the analyte has the same charge as the column surface, then decreasing the relative amount of water will increase the retention of said analyte on the column. 2. If the analyte has the opposing charge of the column surface, then decreasing the relative amount of water will decrease retention, leading to less retention on the column and quicker elution. We've shown an example at the bottom of the slide here, wherein we are using the same negatively charged stationary phase, and the concentration of water is low enough where the doubly charged ions that are positive in charge are coordinating to the stationary phase. And this, in effect, is allowing for negatively charged analytes that are in the sample being analyzed to retain on the column as if it were positively charged in the first place. And by changing the amount of water that may be contained within the stationary phase, one may be able to control which analytes elute first. The method that has been described on the previous slide has become known as bridge ion separation technology, otherwise designated as BIST, given the bridging ions that are utilized in the stationary phase. And for this technology to work, there are certain conditions that must be met beforehand. First, a doubly charged buffer ion must be present in the mobile phase to act as the bridging ion. Second, the buffer's doubly charged ions should be opposite in charge to the charge of the stationary phase itself. Third, there must be reduced water in the mobile phase so as to minimize solvation that might occur. And below, we have shown a general summary of the different kinds of columns that are available. There is BIST A, which is got a surface charge that is negative in nature, and if using an appropriate bridge ion, can be useful for anions. The BIST B columns have a positive surface charge, and thus with an appropriate bridge ion, they can be useful for cations. And BIST itself has a great degree of versatility with separating and analyzing different molecules, with the notable exception of neutral hydrophobic molecules. This universality of this method is not displayed in other popular chromatographic methods, such as ion exchange chromatography or reverse phase chromatography. And now we're going to go over the utility of using BIST to separate and analyze different dye molecules. The ability to analyze and separate dyes in and of itself is important, as many dyes are produced as mixtures of different structural and constitutional isomers. And given that many of these dyes are used as food additives, it's incredibly important that they be free of any possible contaminants that could be harmful to human biology. And their purity needs to be very meaningfully established. Many such dyes actually contain ionizable groups, such as the acidic sulfonate groups shown down below, as well as basic amine groups shown down below here. And these different ionizable groups can participate in strong ionic interactions that make them separable under different conditions. And it's because of this that BIST is a technology that's well suited to separate multi-charged analytes. Now, for our first example here, we want to show that BIST is a useful technology for separating and elucidating different dyes at different retention times. And so for these two examples, we're working exclusively with tartrazine, which is this dye shown down below. It contains 3 ionizable groups shown here, here, and here. 2 of them are sulfonate groups, and 1 of them is an acetate group. And if you were to do a HPLC method using triethylamine as the buffer additive, the tartrazine would elucidate from the column at a very, very low retention time. By comparison, if one were to use the doubly charged tetramethylamino-propane ion, shown here, they would elucidate tartrazine at a much later retention time. This demonstrates that the doubly charged additive in the buffer is crucial and impactful for the BIST technology to do its work. On the right hand of the slide, we have shown the same triethylamine result and TMDAP result, but we've also shown that one could use a different ionic additive, such as magnesium acetate, in the buffer as well to achieve a different retention time, depending on the kind of separation that one is seeking. And on this slide, we're going to show the effect of molecular structure and the number of ionizable sites on the degree of retention on a BIST type column. For all of these examples, we're using a BIST A column, and the buffer that we're using is a TMDAP buffer with a pH of about 5. Now, of these 6 analytes all shown here, the one that will retain the least on the column will be 2-naphthalene sulfonic acid, shown here at the topmost spectra. And this is because it contains only 1 ionizable site coming off of this biaryl moiety. By comparison, toluene sulfonic acid retains on the column for a little bit longer, but it doesn't retain that much better. And this is because even though it does have a slight difference in the aryl moiety, the ionizable site is still only at a count of 1. Now, if one moves down to the acid red 1 food dye, which now contains two different ionizable groups, this compound will elute from the column at a retention time of about 6 minutes, almost double what the previous two sulfonic acids eluted at. And the sunset yellow food dye, which also contains two ionizable sites, elutes at almost an identical retention time as the acid red 1 food dye. By comparison, if one goes down now to the 1,5-naphthalene disulfonic acid, this compound comes out of the column at a retention time of about 10 minutes or so. This is notable because this compound now has two ionizable sites compared to its sibling molecule, which was 2-naphthalene sulfonic acid. So the difference of having two ionizable groups on the same organic structure led to a retention time increase that was almost 5 times the amount. And then if we were to try to elute fast green FCF food dye from these conditions, we would find that the compound, which contains 3 ionizable sites, would elute at a retention time of greater than 12 minutes. So the number of ionizable sites is greatly impactful to the degree of retention on the BIST columns, as is the molecular structure. And on this slide, we are showing a separation of 4 red dyes on a BIST A+ column, but this time using a magnesium acetate buffer with a pH of about 4. The first 2 dye to elute from the column are erythrocene, followed by acid red 1. These compounds elute first because they both contain 2 ionizable sites. By comparison, Ponco 4R and Amaranth elute later in the column, and the reason for that is because they each contain 3 ionizable sites. On this slide, we are showing the separation of 4 different food dye mixtures. It's important to be able to analyze food dye mixtures as they are often composed of different amounts of different dye pigments themselves. And so the ability to ascertain that these food dye are both safe and accurate is valuable. The 4 dye we are analyzing are chocolate brown, royal blue, sunset red, and grape violet. This is all being done on a BIST A+ column and with a buffer that is TMDAP with a pH of about 5. Now, if we first analyze the chocolate brown pigment, we see that we are able to clearly see the sunset yellow dye, the brilliant blue dye, and finally the Ponco 4R dye. It is noteworthy that some dye are more visible when analyzed at different wavelengths than others. If we go on to the royal blue, we see that only the brilliant blue dye is observed. Then, if we go to the sunset red, we see very clearly between 2 different wavelength analyses that we have both sunset yellow as well as Ponco 4R. And finally, if we go to the grape violet, we see that we are able to elute brilliant blue and amaranth. On this slide, we show a comparison between an analysis of a brilliant black dye from one chemical supplier versus a brilliant black dye from a different chemical supplier. As you can see on the spectra below, the separation of the brilliant black dye from one supplier actually did not contain the brilliant black dye at all, which you can see over here as compound 5. Instead, through an analysis at different wavelengths using a BIST A+ column, we found that the brilliant black dye was in fact counterfeit and instead contained sunset yellow, brilliant blue, amaranth, and tartrazine, and that the black color was simply generated by mixing all 4 of these dye together. By comparison, we did run an authentic sample analysis on brilliant black dye and found what the exact retention time on the same method would be for the compound. Of course, the elution order for these compounds also correlates to the number of ionizable groups present in each of the dye. And now we're going to show separation of basic dye using a BIST column. So now we're using the BIST B column series, and we're inverting our polarity charge. Down below, we've shown a separation of the two dye DAPI and Fast Blue B. Here we see that the DAPI is the first compound to elute from the column, and that the Fast Blue B is the second compound to elute from the column, and that clear elucidation of both compounds actually occurs using UV detection as opposed to ELSD detection. On this slide, we wish to show the separation of singly charged basic dye. However, in this case, because the dye are singly charged, they cannot be analyzed using a BIST column, and instead must be analyzed with a PrimeSep 100 column. This is because singly charged molecules don't have sufficient retention, and the BIST column is not effective at separating them. Here we're separating a two-component mixture of ethyl red and brilliant green. Analyzing this mixture at the appropriate wavelengths, we see that the ethyl red elutes first, followed by the brilliant green significantly later. This is due to their hydrophobic interactions, as the brilliant green contains an additional aryl moiety, as well as longer alkyl chains on its different substituent parts, and thus it retains longer in the stationary phase. And on this slide, we show the separation of a three-component mixture of basic dye. Once again, because each of these dye are singly charged, we are having to use a PrimeSep 100 mixed mode column. Of these three, the first compound to elute from the column is pararosaniline, because it contains the fewest alkyl moieties of the three compounds. Crystal violet is the second compound to elute, because now the amino groups, including the ammonium ion, are containing two methyl groups. And the crystal violet lactone is the final compound to elute. This is because even though it contains the same methylated amino groups, it now contains a carboxylic acid group, giving it a greater degree of weight and retention on the column. In summary, the separation of both acidic and basic dyes is a difficult but incredibly necessary chemical process. This is due to their wide use in consumer products and commercial goods. BIST is a technology that can uniquely separate multi-charge dyes by manipulating the concentration of water in the mobile phase that is used. And mixed mode columns developed here at SIELC can also be used to separate singly charged dyes as well. If you have any further questions on any content that was covered in this presentation, do feel free to visit any of these websites, as well as contact us here at SIELC Technologies. We'd be more than happy to receive and answer any of your questions.